Välkommen till dag 2 med SLUs professorsföreläsningar 2026. Mitt namn är Malin von Essen och jag har det stora nöjet att få moderera de här föreläsningarna. När den här föreläsningsdagen är slut, vid lunch idag, då kommer vi över de här 2 dagarna att ha rört oss över ett brett fält av ämnen. Från agrarhistoria till motståndskraftig matproduktion, skogens skötsel, ny kirurgi och innovation. Och ni som är med via länk, ni hittar programmet på slu.se/nya professorer. Och ni kan ställa frågor, precis som de som är här i studion. Och om ni vill ställa frågor så mejlar ni till adressen nyaprofessorer@slu.se. Ni som är här i rummet, ni får räcka upp handen så får ni en mikrofon. Och den är ju en förutsättning för att frågan ska gå ut i sändningen. Så den behöver ni använda. Ja, då börjar vi med dagens första föreläsning. Den handlar om agrarhistoria och jag ber att få hälsa Jesper Larsson varmt välkommen upp på scenen.
Min forskning handlar om naturresurser som skogar, åkrar, ängar, fjäll men också om vatten. Jag är framförallt intresserad av hur människor har förvaltat naturresurser. Och framförallt är jag intresserad av resurser som man har ägt och brukat gemensamt. Och då är jag intresserad av hur man kommer överens om hur man ska förvalta dem och hur man löser konflikter. På det sättet är jag intresserad av normer och regler för markanvändandet. Jag heter Jesper Larsson och jag är professor i agrarhistoria. Jag studerar stora genomgripande förändringar i allmogesamhället, alltså bland vanligt folk, och före det industriella genombrottet i Sverige. Och jag försöker visa hur allmogen själv på genomgripande sätt kunde förändra sin ekonomi och hur du kunde förhandla fram nya sätt att hantera sina resurser. Och den här forskningen, den sker över hela världen egentligen. Och det finns likartade system för hur man har förvaltat naturresurser. Men det är ändå väldigt, väldigt viktigt att man inte tror att man kan föra över kunskap från ett ställe till ett annat ställe. För varje område måste förstås utifrån sina förutsättningar. De resurser som finns där, de människorna som finns där och den historia de har. Jag tycker ju att det är roligt att undervisa. Och i undervisningen så betonar vi bland annat att det som har hänt tidigare i landskapet och i jordbruket har betydelse för vad som händer idag. På sommarkurs i agrarhistoria där jag undervisar, där får varje år studenterna göra ett eget arbete kring en plats som de själva har valt och där lyfta fram hur historien är bevarad. Och på de här redovisningarna är det alltid väldigt roligt att vara med. Och de här redovisningarna blir som små högtidsstunder för mig i min vardag här på SLU.
Det kommer att bli bra.
Ja.
Fint. Välkommen Jesper.
Tack så mycket.
Det framgick tydligt av filmen att du gillar att undervisa. Och du nämnde lite om varför studenter bör studera agrarhistoria. Kan du utveckla det lite?
Ja, men det finns väl många skäl till att man vill studera agrarhistoria. Ett är ju bara att ämnet är så roligt i sig. Och att man kan fascineras av hur kompetenta folk var i äldre tider och hur saker har förändrats. Men det finns ju också en större berättelse om varför historia är viktigt. En är ju att vi har föreställningar om det förflutna. Och de kan vara ganska felaktiga. Och därför är det ganska bra att utmana sina föreställningar. Och tar vi det till en högre nivå, till en samhällsnivå, så kan man väl säga att det finns historier som berättas om varför vi är vad vi är. Och med historisk kunskap så kan du faktiskt ifrågasätta de här. Du kan säga, nej, den här historien stämmer inte. Det har inte varit på det här sättet. Så jag tänker att historia bidrar till vårt kritiska tänkande i samhället. Och agrarhistoria är en viktig del i det.
Om vi bara zoomar in lite på den här kursen då, som du ger. Så undrar att du berättade att studenterna får göra något eget arbete och studera någonting. Vad kan det handla om platser? Vad kan ett exempel vara? Vad är det du får?
Ja, men i den här kursen då så går man igenom stora linjer i agrarhistoria, ganska ytligt ändå. Men vi fokuserar på att inte varje plats, men väldigt många platser har en agrarhistoriskt innehåll. Och de spåren är synliga och kan bli synliga genom att man sätter på sig rätt sorts glasögon. Och då jobbar vi med historiska kartor för att få en kronologi i landskapet. Alltså man tar fram en storskifteskarta, en lagarskifteskarta och så jobbar man med sin plats. Och då väljer studenterna ofta en plats de är bekanta med. Om de inte är bekanta med en plats så hjälper vi med dem med en plats. Och så går man ut och tittar där och man går i landskapet och hittar kanske hängnader, diken, gamla vägar och så vidare. Och jag tycker att varje år så är det liksom att folk ser något nytt i en miljö som de har varit i jättemycket, men de har inte sett de här spåren. Och så talar vi också om hur de kan skötas, hur man kan få dem att synas bättre i landskapet.
Man lär sig läsa landskapet.
Man lär sig läsa landskapet.
Ja, spännande. Innan du håller din föreläsning skulle jag vilja be dig berätta lite om ditt karriärbyte. Du jobbade ju många år i olika roller i kulturmiljövården och bland annat som byggnadsvårdare och landskapsvårdare för att sen kasta dig in i forskningen. Vad var det som gjorde att du valde forskningen?
Jag hade ju 20 år innan jag disputerade i kulturmiljövården. Och alltså jag hade ju jättebra där. Det är ett otroligt roligt jobb att få jobba med gamla byggnader. Jag jobbade i Hälsingland med Hälsingegårdar. Jag jobbade på Öland med södra Ölands odlingslandskap och i Jämtland med fäbodar och annat. Men någonstans var det så att man kom bara till en viss nivå i förklaringar av varför de här landskapen såg ut som de gjorde. Och som ni kommer att förstå av föreläsningen idag så är jag intresserad av det norrländska jordbrukslandskapet och norrländsk boskapsskötsel. Och då var det väl helt enkelt de frågorna som drev mig mot forskningen. Och sen var det så att 1994 hade SLU stiftat ett nytt ämne, agrarhistoria. Och jag blev väldigt intresserad och kom hit som student 97 och läste en termin och sen så gick jag en uppsatskurs år 2000 och sen så kom jag in på en forskarutbildning med tiden. Och då kände jag att jag kunde inte gå tillbaks.
Nej, och nu installeras du som professor.
Precis.
Ja, men låt oss höra din föreläsning.
Ja,
varsågod. Jag ska be att få introducera Jesper Larsson, professor i agrarhistoria. Och Jesper ska berätta om att ligga i framkant hos bönder och renskötare.
Ja, att ligga i framkant, det är att ligga före andra, att ha nya idéer och att också genomföra de här idéerna. Och i denna installationsföreläsning ska jag ta er bakåt i tiden till det vi kallar tidig modern tid. Alltså tiden mellan reformationen och det industriella genombrottet. Och de djur ni ser på bilden här, ren, ko, får och get, kommer att spela en stor roll för den här berättelsen som jag berättar. Men den verkliga aktören, det är människan. Det är genom henne som djuren och markens potential frigörs. Så jag ska tala om 2 grundläggande och genomgripande förändringar i boskapsskötseln. Det är innovativa förändringar där samhällen transformeras och det är genom aktörerna, alltså de vanliga människorna, de vi historiker kallar allmoge. Det är den som genomför det här och i vårt fall är det bönder och renskötare. Så i den första delen ska jag kort berätta om 2 stycken sådana här förändringar i boskapsskötseln. Och det är norra Sverige sa jag. Och i den andra delen ska jag gå igenom 4 faktorer som krävdes för att det här skulle kunna ske. Och hur bönder och renskötare kunde omförhandla sina livsvillkor. Det här är nämligen förändringar som inte sker av sig själv utan det är mycket medvetna processer. Och de här som jag ska tala om idag, en del kan väl tänka sig att det här har väl alltid funnits, men så är det faktiskt inte. Utan det växer fram och det krävs massa beslut av människor. Så den första stora omvandlingen i boskapsskötseln var fäbodväsendets framväxt och etablering. Och fäbodarna är en huvudsaklig sak för södra Norrland och norra Svealand. Och fäbodarna är en periodisk bosättning för sommarhalvåret. Man för djuren dit för att vallas i skogen och de ska beta. Och man mjölkar också getter och kor. Och av den här tunga lagringskänsliga mjölken omvandlar man den till lätta och lagringståliga produkter som smör, ost och olika typer av mesvaror. Och fäboden den uppstod efter den senmedeltida agrarkrisen, alltså de pestvågor som fanns i Sverige mellan 1350 och ungefär 1450. Men de är kom framförallt till under 1600- och 1700-talet. Och på bilden ser vi en sån här typisk fäbod. Här är en väldigt fin bild av Hans Per Persson från Djura, Leksand. Och det här är Björnberget i Leksands socken. Och vi ser vad som är typiskt. Ett myller av djur, ett myller av byggnader och fäbodkullorna här. Saknas på den här bilden gör fåret som också var en väldigt vanlig djur på fäbodarna. Och vad fäbodarna verkligen gör, det är att man kan öka den totala avkastningen från boskapsskötseln väldigt, väldigt mycket. Och varför ska jag komma tillbaka till. Den andra stora omvandlingen av boskapsskötseln var den stora förändringen i renskötseln. Samernas omläggning till storskalig renpastoralism är om möjligt ännu mer grundläggande än fäbodväsendets framväxt. Till och med 1500-talet så hade samernas ekonomi varit baserad på jakt och fiske. Och man hade också små renjordar. Basen var fiske och markanvändningen speglar en ekonomi där man skulle kunna livnära sig på fiske året runt. Jakten var ett komplement, framförallt småviltsjakten, men också vildrensjakten. Medan den lukrativa pälsjakten gav viktiga inkomster till hushållen. Runt år 1600 börjar en stor förändring i hur man håller renar. Och det börjar hos samerna i fjällområdena i nuvarande Norrbottens län. Och anledningen till att man håller ren överhuvudtaget är ju för att det är djur som går att tämjas och att de rör sig mycket lätt i snö och kyla. Och därför användes de från början framförallt för transport. Men hos många samer sker då en övergång från år 1600 och framåt i tiden till storskalig renskötsel, det vi också kallar renpastoralism. Förutom att antalet renar ökar från att man kan ha flera hundra renar, 200, 300, 400 renar, så är det också så att jordarna kan innehålla 1000 och mer renar. Är det också så att renarna sköts mer intensivt, det vallas, det samlas in och man mjölkar renen. För precis som i fäbodväsendet så utvecklas en avancerad mjölkkultur här uppe. Men det som gör samernas förändring i boskapsskötseln mer grundläggande än omläggningen till fäbodväsendet är ju att vi här går från en födoinsamlande ekonomi till en ekonomi som producerar föda. Alltså, vi kan studera en av de grundläggande förändringarna i mänsklighetens historia. Hur vi gick från att vara jägare till att vi blir jordbrukare. Och om någon tycker att jordbrukare är obekvämt att använda det här så kan vi säga att de är livsmedelsproducenter. För det är det man producerar. Och vi kan studera den här övergången med historiska källor. Och då kommer jag in till del 2 av den här föreläsningen. Det är vad som krävs för att det här ska kunna hända. För det är ju grundläggande, hur man så här grundläggande kan förändra sina levnadsvillkor. Och jag ska nämna 4 faktorer här. Där den sista, den om regler och normer, förstod ni av filmen, tycker jag är det mest spännande för min egen del. Men vi måste se alla de här 4 sammantaget. Det första är naturligtvis att man måste ha de naturliga förutsättningarna. Och för fäbodarna handlade det om en enorm utvecklingspotential när man kunde ännu mer använda de stora och djupa skogarna i södra Norrland och Dalarna och fjällnära områden för att använda dem för det rika betet som fanns där. Och för den storskaliga renpastoralismen var det ju de här omfattande fjällområdena som ni ser här på bilden, typ sådana med stora fjällhedar som gav ett rikt bete som kunde utnyttjas. Och när det gäller renpastoralismen så är det också så att det är mycket lättare att hålla samman stora renjordar om man är i ett öppet landskap än om man är i en skog till exempel. Den andra faktorn, det är ju faktiskt att alla samhällen påverkas av samhällen runt omkring en. De omgivande samhällena. Och att fastställa exakta orsakssamband mellan externa faktorer och vad som händer i ett område är ofta lite svårt i historisk vetenskap. Världen är komplex, många saker påverkas. Och jag ska bara ta upp en sak som en extern faktor som påverkar det här och som jag tänker är väldigt viktig. Och den illustreras av den här världskartan från tidig modern tid. Det är alltså att vi går mot den första globaliseringen, den som sker under 1500- och 1600-talet. Och det gör att handeln expanderar. Och i fäbodområdenas utkanter så ser vi då hur järn- och stålindustrier växer upp som påverkar boskapsskötseln inne i fäbodområdena. Och för i renskötselns område så ser vi hur handeln blir mycket mer reglerad och utbyggd. Det anläggs marknadsplatser i varje sånt här samiskt område. Efter den svenska kusten anläggs det städer under 1610- och 20-talet som korresponderar mot varje sån så kallad lappmark. Så Luleå går mot Luleå lappmark, Pite mot Pite lappmark och så vidare. Och med marknadsplatser i Norge längs Västerhavet så skapas ett intrikat handelsnätverk för samerna, vilket gynnar den storskaliga renskötseln. Till de två tidiga faktorerna ska vi då lägga den tredje faktorn, och det är den sociala organisationen, som här får illustreras av denna fantastiska bild av Hans Per Persson igen, över ett fäbodplats uppe i Leksandssocken med korna här framme och kvinnorna här, som alla stickar, ska ni lägga märke till. Så man gör inte bara en sak när man är där, man gör flera saker. För när ekonomin ställs om, då måste man också organisera sig om sig. Och att det var kvinnor som var på fäbodarna, det är ganska känt. Kärt barn har många namn. Fäbodkulla, bupiga, fäbodjänta. Och kvinnor på fäbodar har väckt många tankar och stundtals en livlig fantasi. Ibland en alltför livlig fantasi faktiskt för att säga någonting om verkligheten överhuvudtaget. Men tar vi oss tillbaka till verkligheten nu kan vi säga att fäbodarna hade inte kunnat växa fram om det inte hade varit för en effektiv organisation. Och den organisationen fick man när man kombinerade valljonet med mjölkpigan i en person. Till varje fäbod skickade varje hushåll en person. Och på fäboden samarbetade kvinnorna i olika åldrar. Några tillhörde hushållet, andra var erfarna kvinnor som hyrdes in, och här lärde sig ung av gammal. Och nu ska jag ta mina enda siffror på det här föredraget. Omkring 1690 så vet vi att ungefär 25% av alla kvinnor i Orsa, socken i Dalarna, över 10 år arbetade på fäbod. Kan låta mycket. Men vad det här gör för det här samhället är inte bara att vi får en effektiv organisation för själva fäboddriften. Vi får också att 75% av alla kvinnor och 100% av alla män inte behöver ägna sig åt boskapsskötsel på sommaren. Alltså den mest hektiska perioden under året. När det gäller den tidig moderna sociala organisationen inom renskötseln och hur den förändrades med renpastoralismen är det forskning som pågår just nu. Och jag vill därför inte föregripa några resultat, utan ni får hålla utkik till när de kommer. Den fjärde och sista grundläggande faktorn som krävs om jag tar upp här idag är regler och normer. Alltså det vi kallar institutioner. Det är regler och normer som bestämmer som vad man kan göra och får göra och därmed vad som kan produceras. Rätt utformade ger dem en stor trygghet i markanvändningen. Och vid stora omläggningar av ekonomin, som fäbodväsendet och renpastoralismen, måste de här reglerna omförhandlas och normer för hur vi beter oss, de kommer att förändras. I båda fallen handlar det om hur människor lokalt omförhandlar sina kontrakt. Det är självförvaltningen i de här fallen som möjliggör de här omförhandlingarna. En självförvaltning som är förankrad i högre nivåer. Tydligast blir detta med de lokala domstolarna, där statens representant möter i form av domaren, möter lokalsamhället i form av de nämndemän som finns vid domstolen för att lösa tvister som uppstår. Och på bilden så är det då första sidan av domstolsprotokollet från Luleå lappmark år 1700, och längst ner här finns då nämndemännen listade. Så här är det Nils Andersson i Jokkmokk till exempel. Han var nämndeman vid det här tinget. Så självförvaltningen handlade bland annat om vem som hade rätt att vara på vilken plats, hur länge man fick vara där och var gränserna går mellan olika områden man får utnyttja. Och domstolen kunde också sanktionera, en jätteviktig sak med sanktioner, och de kunde utdöma vite om man gjorde fel. Och på nationell nivå så fanns det ju regler om att om man var skattebetalare här så hade man rätt att använda marken och vatten. Och genom att domstolsprotokollen finns kvar kan vi kartlägga hur mark användes och hur det förändrades över tid och hur människorna samarbetade. Jag studerar främst utvecklingen i Sverige i vid bemärkelse. Men fäbodar och renskötsel finns runt om i hela världen. Och de grundläggande frågorna om hur naturresurser förvaltas och nyttjas och hur människor samarbetar, de är universella. Fäbodväsendet, ett fäbodväsende som det svenska fanns också i Norge och på många andra ställen i Europas bergstrakter. Men övergången till ren pastoralism var heller inte bara någonting som skedde i norra Fennoskandia. Det är en utveckling som skedde över hela den eurasiska kontinenten från runt 1600 till år 1900. Och vad som gör en agrarhistoriker i Sverige lycklig är att vi har ett riktigt bra källmaterial att studera övergången med som saknar motstycke i världen. Tack så mycket. Tack.
Välkomna med frågor, räck upp handen, skicka mejl. Jag börjar med en egen fråga här. Alltså man behöver ju inte konsumera särskilt mycket nyheter i dessa tider för att stöta på de markanvändningskonflikter som pågår idag. Eller hur?
Nej. Markanvändningskonflikter är stora.
Men hur gjorde man på den tiden? Hur löste man markanvändningskonflikterna?
Ja, men det är ju ganska spännande. Alltså de här... nu talar vi om markanvändningskonflikter där man har inte riktigt bestämt vem som har vilken mark, utan man försöker bestämma den så att säga. Och det är ju så ofta. Man hade mark tillsammans på något sätt. Man har rätt att vara på marken och utnyttja den för att man betalar skatt. Och ibland blir det konflikter var gränser går. Och vad man ofta gjorde var att om man inte kunde lösa det mellan personerna så tog man det till tinget. Tinget utsåg personer som skulle medla och man gick omkring ute i landskapet och letade gränsstenar, man letade var de skulle gå, man diskuterade fram och tillbaks. Och vad jag har funnit i min forskning är ju att en sak man säger är att konfliktlösning måste gå fort. Men för sådana här konflikter behöver det inte gå så fort. Utan det är viktigt att man liksom verkligen förankrar de beslut man tar hos dem det berör. Sen kanske inte alla blir lyckliga av beslutet till slut, men man har i alla fall varit med och fått sagt till sitt och man manglade länge. Så domstolen är en sån här väldigt, och att det är de lokala folken. Man nu ser till medlare eller de bor också de här platserna.
Ser du några paralleller till idag?
Ja, jag ser paralleller idag att jag tror att vi försöker lösa för många av de här konflikterna från toppen. Och inte involverar de som det berör. Så kan jag säga utan att behöva säga så mycket mer.
Varsågod.
Tack Jesper för en jätteintressant föreläsning. Man blir väldigt intresserad av att lära sig mer. Jag funderar på, du hade en bild där människor övergick från att samla in mat till att producera mat. Det måste ju varit en väldigt omvälvande förändring för mänskligheten, och det finns ju fortfarande grupper i världen som lever på att bara samla in sin mat och inte producera åt andra. Jag funderade på, när den här utvecklingen skedde då i norra Sverige, fanns det kvar grupper som vill fortsätta att samla in mat och leva på fisket och mat för dagen så att säga. Och de grupperna som ville gå vidare och producera mat då. Och hur fanns de konflikterna, och i så fall hur löste man dem?
Ja, men det kan man väl säga att det finns så att säga. Alla går inte över till ren pastoralism. Många lever kvar ett tag som jägare och fiskare, och framförallt med fiske. Ett sätt man löser konflikten är att man omförhandlar vem som har rätt att få vara på vilken plats. Alltså idag tänker vi också property rights till exempel som ett sätt man har sitt mark, och då äger man allting inom vissa gränser. Men här kan man ha olika rättigheter till bete, till fiske och till jakt. Om man skiljer på de tre så kan man lösa de här konflikterna lättare i det här samhället så att man kan liksom leva kvar. Men när vi talar om de här så är ju att vara ren pastoralism är på många sätt ett mer framgångsrikt sätt att vara. Man får jobba hårdare men avkastningen är högre. Så att på så lång sikt så försvinner ju den andra gruppen kan man säga. Och de finns ju som inte som två grupper som inte förändras. de påverkar ju varandra också så att säga hela tiden. Så att många av de här som kanske lever ett tag på jakt och fiske, de går ju också över till ren pastoralism. Och det uppstår en ren pastoralism också för de som mest lever i skogen. Så att det sker både förändringar i ekonomin, men det sker också omförhandlingar av de regler och normer man har. Eller av reglerna, och det påverkar normerna, så ska jag säga.
Dynamiskt. Varsågod.
Tack så jättemycket Jesper. Jättespännande också att du fokuserar på den nordliga delen av Sverige. Du tog ju upp i din föreläsning att det är de här 4 förutsättningarna för både fäboddrift och renskötsel. Du har också nämnt det här med olika former för konflikter, att det kanske är tuppstyrt utan förankring, och det är ju något som vi ser inom renskötseln speciellt. Både en begränsning av hur mycket renar kan man faktiskt ha i ett område. Det finns ekonomiska intressen som påverkar renskötseln. En av de första förutsättningarna du hade, det var ju också vad har vi för tillgång till mark, naturresurser. Vi har en klimatförändring som verkar påverka ännu mer i den norra delen av Sverige än i resten av delen av Sverige. Hur kommer det att påskynda den historiska utvecklingen framöver för just till exempel renskötseln?
Ja, nu tänkte jag ducka frågan genom att säga att jag är historiker och så sitter på äldre tider. Men vi tror att vi vet ju att temperaturen stiger fortare långt norrut än vad det gör söderut med klimatförändringarna. Och att det kommer att få betydelse, har redan fått betydelse för hur snön fryser och att det är svårare för renarna att komma åt bete på vintern och sådana saker. Och det kommer att få stora fördelar. Jag kan bara säga så här att vi ligger just nu inne med en stor ansökan till en Wallenbergsstiftelse om att titta på klimatförändringar från år 1000 till 1950. För att vi tänker att om vi förstår hur det har påverkat historiskt så kan vi lättare säga någonting om framtiden. Så att jag duckar för att säga för mycket om framtiden. Mm.
Ja, jag glädjer mig åt att du ägnar åt boskap. Det tycker jag är väldigt kul. Och jag har ju som veterinär haft förmånen att besöka några fäboddrifter. Och då kan man väl säga att det var väldigt spännande men lite musealt. Och både människorna och djuren och faciliteterna var liksom lite gammaldags. Så att när var själva, för det tillkom ju som du sa av en anledning, men när började det lösas upp och när var det inte någon mening med det längre? Liksom, när började det försvinna?
Ja, det är ju lite olika på olika ställen i Sverige. Men man kan ju säga att fäbodväsendet uppnår sin peak kanske till 1850. Efter det så går det så minskar det, och det minskar ganska snabbt efter 1890 fram till andra världskriget i många områden. Men de kommer närmare fjällen, områdena, så lever det kvar ett ganska starkt fäbodväsende ända till 1960-talet ska jag säga. Därför de har markarronderingar där uppe som gör att de var beroende av att de har ganska lite inägomark och åkermark, och så de kan inte låta djuren beta hemma, de var tvungna att ha det. Men när för många gårdar lämnar så slutar det där också. Men alltså om man ser fäbodens storhetstid så är det 1600 till 1850, därefter börjar en utgå. Och sen går det jättefort under 1900-talet.
Tack så jättemycket för hemskt intressant som du har berättat om. Jag tänkte om vi växlar från djur till växter, och det har du inte pratat någonting om, men de här lantbrukarna här, jordbruket, de sysslar ju med växtodling också. Och vi hörde lite grann igår om växtförädlingens betydelse över tiden. Hur förhåller sig, alltså vilken betydelse har det fått för till exempel utvecklingen av fäbodväsende och annat?
Nej, men alltså den stora saken som gör att fäbodarna försvinner, dels är det att det blir mer mark i slättbygden, eller inte i slättbygden men nere vid gårdarna när andra lägger ner. Men det andra är ju den stora omläggningen till växelbruk som gör att man kan liksom lägga om jordbruket, och den stora omläggningen till växelbruk kommer efter 1850. Alltså att man kan ta sitt vinterfoder från åkermarken och man kan ha djuren betande på åkermark i en annan utsträckning än vad man hade tidigare. Så det är ju som helt avgörande. Men man sökte sig till fäbodarna också för att det var jättebra bete där uppe. Alltså det står ju i jättemånga källor hur fint gult smör man får till exempel. Vilken hög kvalitet det är på produkterna som kommer därifrån. Men de här djuren som har betat på fäbodarna, de avkastar ju inte lika mycket som djur som man sen har i slättbygderna och där nere. Så att det är många saker som händer. Så att,
Jag har en avslutande fråga.
Ja.
En avslutande fråga. Jo, du nämnde ju att att historieforskning kan ju bidra till att historien skrivs om, såklart. Och då blir jag nyfiken på, och ibland skaver ju det här liksom för människan när men det är inte som du trodde. Liksom. Har du varit med om att presentera forskningsresultat som har skavt?
Ja, men det har jag gjort. I min avhandling så skriver jag ju att, eller jag är helt säker på att fäbodbruket växer fram då under 1500-1600-talet, stora perioder. Och jag var uppe i Älvdalens kommun och presenterade det där materialet och folk blev jättearga. Ja, så när vi var på väg, min fru och mina barn hem i Älvdalen så sa jag så här, jag vågar inte gå ur bilen här, folk är så arga. För de vill ju se fäbodarna att de var från järnåldern och de har alltid funnits.
Ja.
Men jag tänker att det är jätteviktigt att vi försöker förstå vissa typer av jordbruk i en kontext och att de finns under vissa tider. Och det är inte. Sen har det kanske funnits någonting som, men så det har hänt.
Ja,
och jag kan säga att vissa av de här samiska frågorna är väl också så att det inte så att folk har varit jättearga. I review-kommentarer kan man ju få när man har mer självförvaltningsperspektiv att folk tycker att det är fel och att det kan skava så att säga. Men jag tänker att en viss typ av historia är inte färdigskriven. Och då ska vi då vill folk tala om deras åsikt för att ibland är det en kamp i historien också om vilken är berättelsen. För många vill ha en stor berättelse men det finns massor berättelser. Det tänker jag är viktigt att komma ihåg.
Nu måste jag ställa en fråga till på övertid här, för det kom in en här via e-mail. Och då är det en person på Länsstyrelsen i Jämtland som undrar hur ska vi få det traditionella och det levande fäbodbruket och de unika kunskaperna att leva vidare? Om du kan ge ett lite kort svar på det.
Ja, oj.
På den lilla frågan.
Nej, men alltså nu har ju till exempel fäbodarna blivit upptagna på Unescos lista för immateriellt kulturarv. Precis. Jag tror det är jättebra att vi ser dem. Jag tror att man måste vara en jätteduktig mathantverkare om man håller på. Att göra bra ostar. Man kan liksom inte, och smör och sådana saker. Och jag tror att det är det. Och de som kan det och gör det, man behöver inte ha en turistfäbod, man kan bara göra smör och sälja lokalt och det bra, eller göra bra ostar och sådana saker. Jag tror det. Och vara förankrat lokalt. För det tror jag vi ser på mycket sådana här med ostar och sådana saker. De säljs mest lokalt. Om man satsar på en marknad som ligger långt bort så kan de bli jätteglada ett tag. Men ganska fort så hittar de något nytt. En spännande ost från Frankrike, den tar vi istället. Men lokalt så ska man försöka förankra sig.
Tack för det. Stort tack. Då får ni en kort paus till 9:40 då ni får träffa McKie. Welcome back, welcome new participants, and welcome Brendan McKie.
I study life in freshwater, everything from tiny microscopic fungi through water insects and even sometimes fish. Freshwater ecosystems such as lakes, rivers and wetlands don't just look beautiful, they support an amazing diversity of life, and they also do a lot of work for us. They purify water, cycle nutrients, break down detritus and waste, transport water to our houses and agricultural fields, and even provide the setting for diverse recreational activities, things like swimming and fishing and so on. My name is Brendan McKie and I am a professor of freshwater ecology. Unfortunately, in draining the landscape, rivers and lakes are affected by just about every pressure that we are imposing on the environment, whether it be nutrients and pesticides used in agriculture, forest management and land clearance, construction of dams and river regulation for hydropower, microplastics and other kinds of pollutants. And a big part of my research is in trying to understand how these various pressures interact to impact biodiversity and alter the key functions and services provided by these ecosystems. I also evaluate solutions and whether measures such as restoring habitats in rivers really are effective at restoring biodiversity and ecosystem functioning. I think my fascination with freshwater ecosystems began during my childhood in Australia. Some of my best memories are long days spent exploring streams near my house and also rocky shores on the coast. I had this real sense that these were habitats which were just crawling with life, that every time I turned over another stone or looked in another rock pool, there was some new fascinating creature for me to discover. And I still carry this sense of wonder with me, and I want future generations to be able to experience the richness of life and this connection with nature in the same way. My hope is that my research helps to support healthy and functional freshwater ecosystems so that they are able to preserve the level of biodiversity that they should.
Welcome, Brennan.
Thank you.
Water diversion measures date back to at least the Viking Age in Sweden. Large-scale drainage began during the 19th century population growth, and from the mid-19th century, forest drainage expanded to boost timber production on peatlands. And I wonder, what are your thoughts on restoring these wetlands now?
Yeah, thank you for this historical perspective. It's important. It's true that we very extensively drained wetlands and peatlands, not just in Sweden, but throughout Europe and other parts of the world to such an extent that I think it's hard for us today to imagine how landscapes looked 1,000 or 2,000 years ago. Our ancestors, as you say, had good reasons for doing so, but I would like to suggest that we've got very good reasons for restoring some of them now, especially living in this time of climate change where we have an increasing occurrence of floods and droughts. We can think of wetlands acting like sponges in the landscape to soak up some of that excess rainfall that falls during storms. At the same time, providing buffer capacity, storing water, give us some buffering against droughts. And I haven't said anything yet about the biodiversity of wetlands, both aquatic and terrestrial plants and animals and so on. So.
Do you think we're investing in the right restoration measures today, or do we need to rethink our approach?
I think we have a lot of very effective approaches for restoration. There's definitely an imbalance. There are some restoration approaches we use a lot. Habitat restoration, we're increasing wetland restoration, but there are other things we could do more. I think especially of hydropower dams. Hydropower dams are often built on steep river sections because that's where you get the gravity to drive the water through the turbines. And these habitats are very often waterfalls or rapid habitats, high quality freshwater habitats. And I think we could release even small amounts of water to breathe life back into those kind of habitats much more than we do. Another measure I think we could do a lot more of is replanting trees alongside streams and rivers in agricultural landscapes, but you'll hear more about why that's so important in my lectures.
Yeah, we'll come back to that. I know you think it's important to involve local communities in restoration efforts. What could be the consequences if this is not done?
I lecture a lot to my students about the factors which regulate the success of restoration, and I might talk about ecological factors such as the movement of organisms through landscapes and whether they can even reach the restored habitats. But of course, one of the big reasons why restoration fails or never even happens is to do with the level of societal support, that it can water down policies or reduce the ambition of planning when there are key groups that are not supporting action. And I also have examples of where restoration projects that are finished are then vandalized or undermined by people who weren't happy with the restoration project being there. Trees which have been chopped down after being planted
And what could be the reason for that? I know you have an example from Vindelälven.
Well, the Vindelälven example was more of an example of, again, something I'm going to talk about during the lecture is the channelization of streams for timber floating. And during field work there, once I stopped off at a cafe, actually several times, called the Nostalgia Cafe, which was run by this very friendly lady who served us waffles. Her son had been home the day before, but while we were sitting there eating, she took out a photo album and showed us all these photos of her grandfather and great-grandfather working in the timber floating industry, and that she felt there was a certain sadness that we were restoring all these ecosystems. And so I try to think about these issues in a positive sense, that if we can provide good data showing that restoration projects have multifaceted positive effects, not just for diversity, but also for ecosystem services and things that we value, that maybe we can build support for this as an enterprise in the longer term.
Now it's your turn to give your lecture, so please take the stage. And I would like to introduce Brendan McKie, Professor of Freshwater Ecology, talking about fragmented rivers, fading life, the cause for ecological connectivity. That's an old. It's a connected riverscapes, thriving life. The case for ecological connectivity is the right title. I'm sorry.
That's okay. I had an older title which I thought was way too negative, and I want to be more positive. So today I'm going to present to you a case for ecological connectivity. In a few moments' time, I'll tell you about what ecological connectivity is and why it is so important for ensuring that our riverscapes thrive. And then in the second half of my presentation, I will show you some examples from my own research of what we can do to restore lost connections in degraded streams and rivers. But to begin with, I'd like to invite you to join me in contemplating these images of some stream and river ecosystems flowing through what look to be quite pristine mountain riverscapes. What I mean by riverscapes are stream and river ecosystems and the terrestrial habitats that are intimately associated with them. Things like riverbank habitats, floodplain habitats, and so on. And I hope that you, like me, are struck above all by the great beauty and dynamism of stream and river ecosystems. They really are a key component of the way that we experience nature. Stream and rivers are very often described as the blood vessels of the landscape, and indeed in transporting around not only water but nutrients, materials and even oxygen, streams and rivers support the life of landscapes at a very fundamental level. Here what I'm talking about mainly is hydrological connectivity, water-driven connectivity, but streams and rivers are actually connected to the surrounding landscape in very many other ways also. And this is where we get into the topic of ecological connectivity, which can be defined as the linkages among habitats and ecosystems that are mediated by the movements and activities of organisms and of materials produced by organisms. And I'll give you a couple of very concrete examples of what I'm talking about here. And the first of these is the emergence of the adult stages of aquatic insects. The video you're watching here is a mass emergence event of mayflies on a spring evening in Scotland. And mayflies, like most aquatic insects, spend the bulk of their life living underwater as immature nymphs. But when it's time for them to reproduce, they emerge from the water as winged terrestrial adults, and they fly around looking for a mate. And at this point, they are, of course, vulnerable to being eaten by birds, bats, spiders, other terrestrial predators. And here we have a mother yellow warbler, which is feeding its young fledgling, a nice, fat, adult mayfly. But what is the young bird actually receiving here? Well, once again, it is receiving the adult of an insect that has spent the bulk of its life living underwater, and in this case, feeding on algae, such as these diatoms. And this is significant because freshwater algae, and especially diatoms, are capable of synthesizing a type of nutrient which almost no terrestrial organism can produce. And this class of nutrients comprises highly unsaturated, high-quality, long-chain fatty acids, such as the highest quality omega-3 fatty acids. We're encouraged to eat fatty fish or possibly to supplement our diet with omega-3 capsules to ensure that we get these nutrients. And that's because they're essential for the functioning of our brain, other membranes in our body, basic physiological functions, growth and development. And the reason that fish are such a good source of omega-3 fatty acids is because they have been eating the insects which have been eating the algae that produce these omega-3 fatty acids. So this is a prime example of ecological connectivity, a type of nutrient that is produced in freshwater habitats, transferred to land via the adult stages of aquatic insects, and on land it supports terrestrial food webs. The second example that I want to talk about is the example of leaf litter fall. Every year there are gigatons of leaf litter that fall from trees all over the world, and when they fall, they often enter other habitat types, and this includes freshwater habitats. In stream ecosystems, leaf litter is rapidly colonized by litter-degrading fungi and leaf-eating detritivorous invertebrates, such as these caddisflies. And these organisms, in breaking down the leaf litter, release the energy and nutrients that are locked up in that leaf litter for other organisms in the food web to exploit. So once again, this is ecological connectivity in action, a material produced in the terrestrial environment, transferred to the aquatic environment and powering food webs there. The problem that we face today is, of course, that very many of our activities are undermining these fundamental ecological connections. When it comes to rivers, it's easy to think of the example of dam construction, which interrupts not only the free transport of water, but migration patterns of fish and the transport of fine particles, nutrients, smaller sized organisms, which normally would be transported from upstream to support life in downstream habitats and even in the ocean. The second example I have here is of an agricultural stream. Agriculture actually interrupts connectivity in very many different ways, but in line with what I've already talked about today, the obvious element that's missing from this photograph is the forest that used to shade this stream channel. So this linkage between a forest ecosystem and a stream ecosystem in terms of the decomposition of leaf litter is gone. And this is actually a fundamental component of larger scale landscape level carbon cycling. As we push these connections more and more, we begin to compromise the capacity of streams and rivers to support biodiversity and deliver the ecosystem services that we value. This example is a fish kill event in the River Oder in Germany, which is a very highly engineered, regulated river, a very disconnected river. The second example is actually the River Cam in Cambridge in Great Britain. And what you're looking at here is an example of hypercharged connectivity, where excess inputs of organic pollutants have stimulated an algal bloom. And the River Cam is, of course, an iconic river in England. It's famous for student punting trips, but I don't think many people are going to want to go punting on the river when it looks like that, and I leave it to you to imagine how it might smell. So what can we do about these kind of problems? Do we have management measures that can address degraded ecological connectivity? One activity which we have been investing in increasingly over recent decades is habitat restoration. And we have a couple of examples here for rivers. We can use heavy machinery to replace boulders into rivers, or explosives to remove dams which are no longer needed. These kind of restoration measures undoubtedly change the way the river ecosystem looks, but the question is, do they have positive effects on biodiversity, and can they restore lost or degraded ecological connections? And for the remainder of my presentation, I'm going to show you some results from my own research which address these questions. And the first example that I'm going to talk about is habitat restoration in channelized stream ecosystems. It's possible to find these kind of streams throughout Sweden. They date from the so-called timber floating era, before Sweden had an extensive network of road and rail. The main way that we transported timber from inland to the coast was by floating sawn logs along streams and rivers. To do this, we needed to modify stream and river habitats extensively. And in particular, what we did was we blew up boulders, which used to be a very prominent habitat element in streams and rivers, and used the fragments to create these kind of canalized banks. And what we're left with here is a very simplified ecosystem. There's not a lot of habitat diversity here, and these systems are also not very retentive. Water just rushes through them because that's what they've been engineered to do. Starting in the 1980s, but gathering pace especially since the year 2000, there have been increasing efforts to restore habitats in these channelized systems. This starts off with breaking up these canalized banks and then replacing smaller sized stones and even really large boulders, and also knocking down trees to try and recreate woody debris dams. And there are some other things like reopening closed off side channels. Once again, this kind of restoration changes the way the ecosystem looks, at least to our eyes, but the question is, what does it alter ecologically? Well, one of the effects that we can see almost immediately is that the systems become a lot more retentive. All of these extra habitat structures slow down the flow of water and entrap materials such as leaf litter. That means that the leaf litter has a chance now of being decomposed locally, close to where the litter fell, instead of being washed downstream, perhaps to accumulate at the bottom of lakes, where decomposition processes tend to be more anoxic. We also see some important changes in the composition of biological assemblages, and here I'm going to focus on insect species that eat leaf litter. In the channelized systems, we tend to get a lot of stoneflies. These are relatively small sized and they do eat leaves, but they do it more by scraping at leaf litter surfaces rather than really chewing the leaf litter, so they break down leaf litter relatively slowly. In the restored ecosystems, this greater habitat diversity starts to see us getting a wider variety of leaf-eating insects, including litter-decomposing caddisflies. These are much larger sized, they've got really robust mouthparts, and they really tear the leaf litter apart. So leaf decomposition goes faster when you have caddisflies present. And we have been able to show that these changes are associated with increased rates of leaf decomposition. We study leaf decomposition very simply by enclosing leaf litter in these kind of mesh bags, leaving them out in the stream for 6 to 8 weeks, retrieving them and seeing how much mass has been lost. And we've been able to document that leaf decomposition goes faster in the restored habitats than the channelized habitats. And putting all of this together, greater retention of leaf litter locally, faster decomposition, says to me that we have succeeded in enhancing the linkage between the forest and the stream ecosystem, mediated through decomposition of leaf litter, which is a key component of landscape level carbon cycling. Okay, the second example I'm going to talk about is the planting of trees in the riparian areas alongside agricultural streams. Riparian area simply refers to the riverbank area and then some distance out from the stream channel. What happens when you plant trees here and they begin to grow and mature? Well, one of the things which obviously happens is the streams start to become shaded. And this helps to regulate temperature. It can protect stream ecosystems during summer heat waves. The roots help to bind the soils, and this reduces erosion and sediments. And trees can also take up some of the excess nutrients that we add to the landscape. Furthermore, the tree roots will begin to reshape the stream channel and increase habitat diversity. And we've been able to show that these improvements in habitat quality positively affect multiple aspects of biodiversity, and in particular affect the biodiversity of some of the more environmentally sensitive species. So in other words, having sections of stream in agricultural landscape with these riparian forest buffers helps to create small islands of high quality habitat, which can support environmentally sensitive species. But what about ecological connectivity, which after all is the main focus for today? Once again, it is informative to look at variation in community composition. And here I'm looking at invertebrates because they're my favorite organisms. What we find in these unbuffered stream systems is that they tend to be dominated by environmentally tolerant species of snails and worms. And these are organisms which play important roles in freshwater ecosystems, but they're not insects. They're never going to sprout wings and fly away from the stream. And as such, they're never going to be a big component in the diet of most birds and bats. We do get aquatic insects, but they tend to be smaller sized, weak flying species, such as this non-biting midge. Spiders living close to streams will catch thousands of these animals and happily eat them, but they're doing it close to the stream channel. These animals are not going to fly very far from the stream, so in that sense, the aquatic to terrestrial linkage is weakened. In the buffered stream sections, the improvements in environmental quality start to favour insects over snails and worms, and in particular insect species with larger sized and strong flying adults, such as damselflies and caddisflies, animals which are going to fly much further into the terrestrial environment. This is a photograph here of some sticky traps that we put out in some areas of riparian forest, and you can see these nice big fat caddisflies which are very attractive as a food resource for birds and also for ground hunting spiders such as wolf spiders. And we can actually quantify the improvement in connectivity here even more directly by once again looking at our old friends, the omega-3 fatty acids. And what we've been able to document is that concentrations of long-chain omega-3 fatty acids in riparian spiders are linked to the availability of strong dispersing insects from buffered stream sections. So in other words, riparian buffers increase the access of riparian spiders to these nutrients produced in aquatic habitats. So I think that I've been able to show you that some restoration measures, which in theory at least should be relatively simple, replanting trees, putting rocks back into streams, can improve multiple aspects of ecological connectivity. A lot of our restoration work that we do today is based on a paradigm that we replace or regenerate particular habitat features to support biodiversity. But regenerating habitats alone doesn't guarantee that ecological connectivity will be improved. And I would suggest that ecological connectivity needs to move more to the heart of the way we think about restoration. Or in other words, we shouldn't only aim to restore biodiversity in habitats, but also these key connections that link habitats together, support life in landscape, and the delivery of ecosystem services. I'm not sure how many other people did this, but I'm going to do this and thank all of those who supported me in getting to this point in my career, going along back, earlier supervisors and mentors, all the students and postdocs I've been lucky enough to work with, and especially Andre Freiner and Frank Burden, whose results I presented a lot today, and of course my family and friends. So, and all of you for listening. Thank you.
Welcome back, and thank you very much for a very interesting presentation, also a very beautiful presentation. And you're welcome with questions, of course. In your last slide, you said that replanting trees and replacing rocks in rivers are simple restoration measures in theory. Why do you consider them simple, and why did you emphasize in theory?
Simple in the sense that what could be easier than planting trees, getting the heavy machinery to put rocks back into streams might require a little more planning, but it's also relatively simple. There's no high technology involved in these methods. The in theory part comes because, of course, it's more complex than that. You want to plant the trees in areas where the trees are going to actually be able to survive. But it's especially more complex because of this interaction, again, between science, action, and society. So planting of trees, I will say until I'm blue in the face that planting trees alongside agricultural streams improves the environment a lot. But it's not something that's done very much in Sweden. There are arguments put against it. It's not supported at the highest governmental levels. One of the arguments is to do with food security, that if we end up in a time of famine or war and there's a need to beef up agricultural production quickly, we don't want to have potential land covered with trees. And in some ways, these arguments are hard to argue against, especially at the moment. But as always, we hope that if we can present restoration effects in a broader sense, not just one aspect of biodiversity, but ecosystem services, habitat improvement, improve conditions for people, then maybe we can still make a case that this is a good thing to do.
Thank you. And how broad should this riparian zone be to be helpful for the organisms in the river?
Yeah, I mean, the complicated answer to that question is it depends which organisms you're talking about and which aspects of the ecosystem you're talking about. In a project that we had where we looked at this, we took a very pragmatic approach and looked predominantly at buffer length, because if you're a landowner, it's maybe easier to think of giving up a longer length of land along a river than a broader length.
Makes sense.
And I mean, we start to see improvements with buffer lengths of around 100 meters. So it doesn't necessarily have to be a lot, but it's actually the network of buffers that's more important, to have frequency of forested areas maybe rather than just one very long.
Well, the connectivity,
yes,
in
exactly.
the landscape.
Thank you, Brandon, for inspiring presentation and talk. I feel really connected because I took supplement omega-3 this morning.
Okay.
Yeah, but I have a question related to that. So I think it was a great example. But how much do we know about these biomolecules and that are essential for other organisms in the ecosystems? How they are produced, and what's the knowledge level there?
Yeah, there's another guy in my department who's the real expert in these questions, and that's Danny Lau. So I can give you an answer, but you should probably talk to him. So I mean, just with the omega-3 fatty acids, there are short-chain fatty acids which are produced by terrestrial organisms, but the really long-chain ones are produced predominantly by algae. They're definitely important for all vertebrates, for membrane functions and so on, but they're important for all organisms. They play these roles in transport processes across membranes and so on. But I'm not a physiologist who works at that level. So.
It's okay.
Yeah.
When you talk about planting trees along the streams that crosses agricultural land, we know today that we have more and more cases of climate change-induced floodings. So how would that be positive to sort of facilitate taking care of the water that comes down, or would it even be negative in that sense?
I think this
How much do we know?
I think this is one of the great unresolved or great challenges when it comes to communicating and understanding the roles of trees in agricultural landscapes, because there is a perception among some landowners that planting trees can end up with a more kind of spreading form of stream channel, which increases the flooding risk locally and maybe at the wrong time of year when they are wanting to plant their crops and so on. I'm not a hydrologist. I've talked to hydrologists about this issue, and they say that this shouldn't necessarily have to be a problem, but it's absolutely the sort of thing which needs to be modelled and investigated. I think trees are a component of what are called natural water retention measures, including things like wetlands, reducing the number of hard surfaces in the catchment roads and so on, all things that can help to retain water in the landscape. But there are tensions there as well. Forests take up water, and then maybe agricultural landowners feel like there's less water for them. So this is a very complex problem.
Thank you very much, Brian, for a very interesting lecture. I noticed from the video you're very much boots in the water, measuring things on site. In your area of research, how much is DNA barcoding becoming more established, so where you take samples of water and use sequencing technologies to find out what's actually living there?
Yeah, that's moving extremely fast. I think that I've only just started using DNA barcoding myself to identify fungi living on leaf litter, which otherwise are very time consuming to identify in the laboratory. But there is a section in my department that is focused very strongly on this and comparing the results you get from eDNA-based samples from traditional taxonomy, how well they match and so on. There are some challenges when it comes to stream ecosystems, the biggest challenge being that the water is moving. So if you take an eDNA sample here, what area of stream ecosystem is it telling you about community composition? And if, for example, you've got forest upstream in a clear cut here, are you really getting information about the forest upstream? But it's very active, and we're applying for lots of money to work with this. Yeah.
Thank you, Brendan. Is it your research that is the base for the restorations, large-scale restorations that they are doing, or is the restorations large-scale experimental study that you use?
Well, the particular timber floating restoration example, that was an EU LIFE project in Vindelälven's catchment area. And for those of you who don't know about EU LIFE projects, they fund restoration actions, but they don't fund much monitoring and assessment. But for us researchers, this is a real opportunity to jump on, and we can find a little bit of extra money and take some samples. And, of course, what we hope is that our results will improve future restorations. And we have now the EU restoration law, which is setting mandatory targets for nations to restore habitats and species. But we also have a problem, which is that we have done a lot of restorations, but not necessarily evaluated how well they worked or the reasons they succeeded or failed. And so when people come to me and ask what are the best combinations of restoration measures to be applied here, then I don't necessarily have the answer to that. So this need for ongoing research is definitely there to ensure that we don't make the same mistakes again and again, but instead improve successfully. I'm not sure if that answered your question or not, but no.
And the last question, the final question. What you mentioned the EU nature restoration law. What do you think it will mean for this kind of research?
Yeah, I mean, it's an example. The EU restoration law is a wonderful opportunity to rehabilitate habitats and networks of habitats all over Europe. But we want to do it in a good way. And we have all of these large knowledge gaps about, for example, spatial planning of restoration measures. How do they interact with one another? As often when the EU passes a new law like this, there's a lot of funding calls that come focused on restoration, and that's good. You could think it's a little bit after the fact, but such is life. The problem is a lot of these projects, of course, are going to be 3 or 4 years, whereas what we really need to do is to be able to follow restoration projects for decades to answer the all-important question about when can we expect to see a positive effect from restoration, because it's usually a mistake to sample the year after a restoration project and expect to see a dramatic improvement in biodiversity, even though we want those kind of quick answers.
With that, we say thank you very much.
Thank you.
Nu blir det paus till 10:40 och då är ni välkomna tillbaka och få möta Valentin Picasso. Welcome back and welcome Valentin Picasso.
Well, I grew up in Uruguay and I used to visit my family farm where there's a lot of grasslands and a lot of different grasses and animals. And I fell in love with that ecosystem that produces both food and also biodiversity. And those ecosystems are globally under threat. And I quickly learned that to protect those ecosystems, we needed to do research documenting what are the benefits and what are the challenges that are facing. I'm Valentin Picasso, and I'm professor of cropping systems. My research aims to develop cropping systems that are more sustainable and more resilient to climate change. I try to design agroecosystems that are both more perennial and more diverse, just like natural ecosystems are. I have focused on forage systems like grasslands and leys that are very important for global protein production. I've also researched silvopastoral systems that combine trees and cattle and are very important for mitigation of climate change. And finally, I focus on perennial grain crops, which are new crops that are perennial and produce grain and forage. You plant them once and you can harvest them for multiple years. And because they are perennials, they cover the soil year-round, they reduce soil erosion, they are good for protecting the water, providing habitat for biodiversity and mitigation of climate change. I do think that agriculture can be an important contributor to the solution of the main problems that humanity has. So by reducing hunger and increasing food security, but also mitigating the environmental problems and providing a society that is more fair and more sustainable.
Welcome, Valentin.
Thank you.
We heard in the video that the aim of your research is to develop new, sustainable and more resilient cropping systems. And as I understand it, you see a need for a fundamental change of agriculture and towards a more diverse and perennial crops. What signs indicate that this transformation is already underway?
Well, first, I think both farmers and industry and policymakers even have realized that we need to reduce the environmental impact of food production. So I think the awareness part, the part of understanding that's the need, it's a good sign that we're moving into that direction. Some people think that we can do small changes like reducing fertilizers or using cover crops. But some of us think that we really need the more radical transformation, changing the roots, having perennial crops. When I was in graduate school, the idea of perennial grain crops was like a futuristic idea. Today, we can eat the food and drink drinks from products of perennial grains. That shows that the transformation is ongoing. Mm-hmm. Initially, there were a few people really looking at this, and now there's really has moved outside the core group of researchers, and there's more and more people looking into all these crops. So I think those are really hopeful signs that we are seeing a transformation ongoing.
Maybe you will give us examples in your lecture, but could you mention one or two examples of what you could eat or drink that is from perennial crops?
Well, there's for instance with kernza, which is one of these perennial crops. There's kernza beer, there's pasta, there's pancakes, there's flour. So they're in North America that those are available, and I have drink and eat those. Yeah.
And you liked it?
I love them. Yeah.
Well, in what ways does research itself need to change in order to support such a big transformation?
Well, I think really research needs to be more systematic. We need to be more transdisciplinary. researchers have been taught to know a lot about a very small thing, and sometimes that thing becomes irrelevant. So we have to be brave and look at the big picture, look at the system, look at the context, be able to explore the interactions between the different components of the cropping systems. And we need to work outside our disciplines, like with other researchers, but also outside academia with farmers and policymakers and industry and society in general, in order to develop research that's usable and pertinent.
And how do you do this in your research? How do you take society into your research?
Well, the first thing is, even though it might feel much easier to just have your project plan meetings with your colleagues, we need to spend the time to invite all these other stakeholders from the beginning. We need to, okay, are we going to work on a research project? Okay, invite the farmers and the actors to discuss what are the goals, what are the needs, listen, frame the problem together. And if we start from the beginning together and we work together over the process, then the result will be much more relevant for the solution that we're looking for.
Please tell us more about your research.
Thank you.
I'd like to introduce Valentin Picasso, Professor of Cropping Systems, talking about how diverse and perennial cropping systems can give sustainable and resilient food production.
So today in this lecture, I want to tell you why sustainable and resilient food comes from diverse and perennial cropping systems. So food comes from crops or animals that are part of a cropping system. And those cropping systems conform landscapes that eventually define how sustainable our food production is. For instance, if we're eating meat comes from animals that may be grazing on a grassland and may create landscapes that are very diverse and very beautiful. When we have, for instance, when we eat bread or beer or kanelbuller, those come from annual cereal crops like wheat or barley that come from this type of cropping systems that may develop this type of landscapes. Now, these different types of landscapes affect the sustainability attributes. Are we protecting the soil or are we eroding the soil? Are we protecting water? Are we conserving biodiversity? What is the impact on climate? Are we having systems that are productive and profitable and fair socially and economically? Are they stable and resilient to climate change? So all these are the sustainability attributes that are defined by the core of how we manage these cropping systems. Now, what are cropping systems? Well, cropping systems are systems that have a lot of components. Of course, the crops, livestock, the farmers that manage the crops, the soil, but also other elements of biodiversity like pests, diseases, weeds, natural enemies. And there's a lot of interactions. All these components interact. The crops may feed livestock, take resources from the soil. Pests may affect our crops, but also natural enemies of the pests may reduce the problems. And all those interactions are very important to understand the functioning of the system. And as a system, they have inputs, things that come from outside, like rain and sun, that are natural inputs, but also human develop inputs like pesticides, herbicides, fuel, fertilizers, with the goal of some outputs that are either desirable outputs like food or services, but also negative outputs like soil erosion, nutrient leaching, and greenhouse gas emissions. So the main point I'm trying to make here is that cropping systems connect food production with sustainability. And there are two dimensions of cropping systems that are, I think, relevant to define those sustainability attributes. One is how diverse the cropping system is. That means having several crop species over space or over time. And the other one is how perennial they are. That means having crop species that live for several years. So I'm going to tell you three stories to give an example of how these perennial and diverse cropping systems can be more sustainable. My first story is from the grasslands in Uruguay, where I grew up. And in those grasslands, there's beef that is produced in 3 different types of systems. Some of the beef in Uruguay is produced directly on native grasslands that, as I mentioned, are very diverse, a lot of different species and very perennial. Some are produced under sown pastures, or what we call here lays, that are mixes of forage species. And some are produced in feedlots, which are fed grain from annual cereals in a sort of confined or open-air way. Now, when we study the environmental impacts of those 3 types of systems, we see that adding up for the carbon footprint, the fossil energy use, soil erosion, nitrogen, and pesticide toxicity, which is a broad way of evaluating environmental impacts, the beef that comes from grasslands have much more lower environmental footprint than the beef coming from a feedlot. That means it's not the beef that is the problem, it's the cropping system where it's produced that may affect whether it has a high or a low environmental impact. So that's the first story. And why is that? Well, when we have crops, they have a lot of problems. When we have grasslands, they really reduce all those components and the need for inputs and increase the ecosystem services and reduce the negative impacts. Now, the second story has to do with these other long-term features of cropping systems. This is a graph that shows the yield of a crop over time. And the productivity is one thing that we often care in food production, which is the mean or the average of that production over time. And we want higher yields and we don't want lower productivity. But also we care about stability, which has to do with these fluctuations over that. And we want systems that are more stable over time. But also, when there is a big crisis, like in this year that the yields were very low, we want systems that don't fall too much and that are resilient, that maintain the yield even in the effect in the case of big crisis. So how do we study stability and resilience of long-term cropping systems? Well, one of the most important things is we need long-term cropping system experiments. Sweden is a country that has 60 ongoing long-term cropping systems experiment. It's a jewel, a treasure of Sweden. Some of them have been going on from the 1950s. And they are very important for these kind of questions. This is a picture of one from where I did my research before in the US that compares maize and maize soybean and more diverse and more perennial systems with lucerne. So they have a range of diversity and perenniality. And when we study these systems, we found that the more perenniality that was in the crop rotation, the more stability of the output. That means that perenniality is important to provide stability. And when we look at the diversity, how many crops were in those rotations, how many species were in that, the resilience to drought also increased with diversity. That means that in order to have a more resilient system, we need more diversity in the system. So again, this has to do with the interactions that happen in the system. When we have diverse and perennial rotations, we improve the soil and the natural enemies, which reduces pests and disease and weeds, and that reduces the need for inputs and increases the environmental benefits of the system, reducing the negative impacts. So, not only cropping systems connect food and sustainability, diverse perennial systems are sustainable and resilient. Now, that's my second story. The third story I wanted to tell is this. So it's pretty obvious so far that as long as we have perennial forages in the rotation, we're going to reduce the environmental impact of our cropping systems. Now, the problem is that we, humans, are grass seed eaters. We like to eat cereals. We eat maize, wheat, rice. Only those 3 crops account for more than half of the calories of the global population. So unless we find cereals that can be sustainable and that can reduce the environmental impacts and be resilient, we're not going to make the big changes that are needed. So this is a picture of annual wheat.
In the fall, this is the above ground, and this is below ground, the roots. In the fall, in the winter, in the spring, and then in the summer after harvest, there's no crop. And that means that when you are looking at it from the window of the train in the landscape here, or when you look at it from above, there's several months that there's green crop when the wheat is growing, but there's other months that you only see bare soil, bare ground. And bare soil means that there's going to be soil erosion when it rains, that there's going to be nutrient leaching, and we're going to lose nutrients and pollute water. And it also means that we're going to have greenhouse gas emissions and lose carbon. That's a natural thing that happens when we grow annual crops. Now, when we put a perennial crop here, and this is intermediate wheatgrass, side by side with wheat, you see the amount of roots that it has and also the continuous green over time. When we have a continuous cover of the soil over time, and we have roots that are absorbing the nutrients year-round, then this soil cover and the roots mean better soil health, mean clean water, and mean fixed carbon that reduces climate change. So I think this is a very powerful picture that tells us why we need perennial grain crops. The first one is Kernza. Intermediate wheatgrass is the species name. Kernza is the commercial name of the crop, and is the first perennial grain and forage crop. That means that we can grow it, we can harvest the grain, we can, with that grain, make flour and pancakes, or we can fermenting and do beer. And then also we can harvest the forage after harvesting the grain. So in the same season, we can harvest grain, and then later on we can harvest forage. And the most important thing is that the next year it grows again, and we can again harvest grain and again harvest forage without the need to replanting. And we can do this year after year after year. Now, of course, because we want diversity in our system, we can intercrop or grow it together with some legumes, like in this case, Kernza is growing with red clover or Cura clover or lucerne, and that gives diversity to the system. The legumes provide nitrogen. That brings a good thing for fertilization. And basically, you already understood this, I think, when we move to perennial grain systems, all the benefits, reducing problems, reducing inputs, increasing services, and increasing and reducing the environmental impacts. So, but there's also other perennial cereals that are important to mention that are in development, perennial wheat, perennial cereal rye, perennial barley that is domesticated and bred here in SLU. And there's also other perennial grain crops like perennial rice that is a cross between annual rice and a perennial relative and has the same yields as annual rice. You can harvest it for 4 years. They have done it in China for 4 years with the same yields. So that's a really great success story. There's also perennial sorghum, perennial sunflower. And here in Sweden, one of the projects we have is the one we call the Viking project, which is validating the introduction of Kernza in the Nordic Baltic region, the best named project ever. And where we have Sweden, Norway, Finland, Estonia, Lithuania, and Denmark testing this new crop, looking at the grain yield, at the forage yield, and the products. So perennial grains, I think, are the future of sustainable food production. Now, how are we going to get there? Well, let me try to summarize then. Cropping systems that are diverse, reduce weeds, pests, and diseases, reduce the need for inputs, protect biodiversity, increase resilience, and perennials reduce soil erosion, reduce water pollution, fix carbon, protect biodiversity, and provide stability. So I always tell my students, think about in these two axes when you're thinking in a cropping system. Look at diversity, the number of species, and perenniality, continuous soil cover, and think that when we're moving from these less diverse annual monoculture systems to more diverse perennial and grasslands and polycultures, we are going in the direction of ecological intensification, improving sustainability, improving resilience. Now, how do we do this? How do we move from these annual monocultures to those diverse perennials? There's always barriers, socioeconomic and policy barriers that keep this system in place. And in order to solve this, it can be done only with science and agroecology. We need transdisciplinary teams where we work together with different disciplines, with different actors in framing and thinking about these problems together. So transdisciplinary teams are central. Now, also, we cannot do it as an only country. We need international collaboration. Of the 17 Sustainable Development Goals, the 17 is we need partnership for the goals. This is a map that shows some of the countries where we have active collaborations right now on these projects. And this is what we really need, transdisciplinary teams and international collaboration. So as I was preparing this final talk, I was reflecting about what it means to be a professor, and I went to the dictionary and I said, well, a professor is somebody who professes. Okay, great. And so what is to profess? Okay, to profess is to declare openly or freely, to confess one faith in, or to teach. So as a new professor here at SLU, I want to profess that cropping systems connect food and sustainability, that diverse perennial systems are sustainable and resilient, that perennial grains are the future of sustainable food, and that transdisciplinary and international collaboration is key to achieve these goals. And finally, that I want to profess that we really need science and education for a sustainable life. Thank you very much.
Thank you very much, Valentin. So interesting and convincing. When leading, and you're welcome with questions. And those of you who participate online, you can email your questions to nyaprofessorer@slu.se. I have one question. When leading large, complex, transdisciplinary teams, what have you learned about what make these collaborations succeed or fail? You mentioned to be close to the farmers and the actors, but can you tell us something more?
Sure. Transdisciplinary science is team science. So the things that work for a good team work for transdisciplinary science. So think about a football team, for instance. You need that the players have played together for a long time. You need time sharing, getting to know each other, and communicating and understanding the language of each other. And you need, for that, it needs time to build those teams. There's no way you are going to do it very fast. You need a lot of years. And you also need diversity. You need some people who are running in the mid-camp, and some people defending, some kicking the goals. The same thing happens in a transdisciplinary team. You need diverse group of people, diverse disciplines, diverse expertise, diverse genders and career paths and whatever. So in a way, when you think about it, success in transdisciplinary teams, just like success in cropping systems, requires perenniality and diversity.
Thank you. Yes, please. Yeah.
Thank you very much for a very interesting presentation, Valentin. And of course, I was delighted to see the role of animals in there. And then also in your presentation, as we heard yesterday, there's often we talk then about ruminants, either cattle or sheep or other types of ruminants. When you think about this sort of integrated systems, what role do you see for other food-producing animals like monogastrics, pigs, or aquaculture species? Where do they come into this picture?
That's a good question.
What is monogastric?
Monogastric means they have only one stomach, like humans, but that includes like pigs and chicken.
Okay.
So, so I mostly have worked with ruminants because I do think they have a very important role on protecting grasslands, and grasslands are really important ecosystems to conserve. When we think about the monogastrics, they eat the same things usually as humans do, and so there's this issue that sometimes comes with like the competition for food and animals. But I do think there's a role of monogastrics too in the production. They are much more efficient at food conversion than the ruminants too. So I think again, diversity is important. We need different types in different contexts in different places. So every animal and crop have a role in having a sustainable system.
It seems like it's a big change going from harvesting a perennial to an annual crop. So would there be, what type of challenges? You mentioned socioeconomics. I know that farmers need to invest quite a lot in machinery. Would you need completely different machinery? And what are the others for the farmer?
Yeah, that's a good question. So really the machinery is something that we already have, like as farmers already harvest seeds of grasses that are smaller, and so there's not a lot to I mean, of course there's always things to tweak in the technology and adaptations of that, but it's not a big hurdle. The main thing is that we need to make these systems that are more environmentally friendly. We also need to make them profitable, right? And for that, the tool that we have always used to make cropping systems profitable is policies that support some types of systems versus others. And so I think the main hurdle is to promote policies that would reward the farmers for the environmental benefits they are producing with this, and support this type of transformation, really.
Can I just ask you, as a professor, what can you do to increase the likelihood that the knowledge you produce actually reaches and influences policymakers then?
Well, I mean, like it's basically coming back to transdisciplinarity. That's the first thing. I mean, it's like involving them from the beginning in the project. It means paying attention to what they are asking, and also learning that the times of the policymakers and the researchers are different, but we need to communicate. A research project usually takes years to be developed and to find the results and to analyze them and to publish, whereas policymakers say, tell me what I need to do tomorrow because I need to make a policy and make a regulation, right? So we need to learn and develop these long-term relationships so that we understand that some things take more time and some things need to be acted immediately. So that is important.
Relations. Yeah. We have a question from an online participant. What needs to be improved in Kernza to make it more attractive to farmers today?
Yes, well, really the bottleneck today for farmers to actually grow Kernza, we have farmers that ask us, okay, I want to grow Kernza here in Sweden, and is that in Europe it hasn't been approved yet as a new food. And so that is one thing that needs to happen, and because the regulatory process hasn't really ever approved a new cereal crop, because we've eaten cereals forever, the process is a little cumbersome and it's not very easy to navigate. But once that is done, which we're very hopeful that it will happen this year, farmers will be able to grow it. And then because it's this dual use, although the grain yield of Kernza is still low, and of course it needs to improve, and that's what plant breeders are doing, are working to improve the grain yield, the forage production and quality is really good. So with those two sources of income, the grain and the forage, it would work.
Yeah, exactly. We have maybe a final question.
Thank you so much, Valentin, for a very inspiring lecture and also with a very clear message. I was interested in, you showed the diagram regarding the different kind of grasslands. And of course, the feedlots is those who is the absolutely most polluting system of actually producing beef. But I also noticed that the natural grasslands, the carbon footprint was higher for that group. And I wanted to ask you why, and also do you have any idea how we could decrease that carbon footprint on natural grasslands?
Yes, it's an excellent point. So the growing cattle in grasslands usually has a lower rate of putting weight on. So it usually takes longer. And the carbon footprint of the beef production depends on how long it takes to produce the beef, because it's the ruminants produce methane and all that. So one way to reduce that carbon footprint is to manage the grazing in a more efficient way so that the cattle eats more frequently fresh grass rather than older grass, and that speeds up the process and reduces the carbon footprint. Okay, so that's one way of reducing the carbon footprint. Anything that increases efficiency in reproduction or in nutrition is going to reduce that carbon footprint. Now, it's important to note that although climate change and carbon footprint is very important, all the other environmental impacts are also very important. And so sometimes we have this fixation that, okay, we need to focus only one thing, but we really, again, we have to be brave and look at the big picture and see all the environmental impacts. And we really need to reduce them all. That's the main thing.
Short comment.
Actually, a kind of trade-off and also a weighting of which is one important.
Yes.
Start to focus on.
Yes.
Thank you.
And yesterday we learned that grazing could be more efficient in the night. Mikaela Lindberg told us about that. Anyway, a warm thank you, Valentin Picasso.
Thank you very much.
Ja, och då blir det paus till 11:20 då Levi Yant ger sin föreläsning och det är då den sista av årets professorsföreläsningar. Så välkomna tillbaka om några minuter. A warm welcome to Levi Yant.
I've always been interested in science, and I love being in nature, and I love seeing how species, any of them, birds, plants, insects, live in nature. That we can now apply technology to understand the solutions that evolution comes up with in response to known challenges that we can apply finally to our crops is a perfect mixture. So it's a great privilege to wake up in the morning and be able to do this. My name is Levi Yant. I'm professor of genetics at SLU Uppsala. We studied the solutions that nature has come up with to extreme challenges for the cell. Occasionally, a cell in nature produces twice as much DNA. This presents extreme challenges inside of the cell to organize this DNA and to put it in the right places. Usually this is fatal and a cell does not survive, but occasionally the cell adapts quickly and then uses the extra DNA to adapt to changing environments. Our work focuses on those solutions and understanding them using large-scale DNA sequencing data. We can take this information learned from nature and apply them to our most important crops to make them more resilient in the face of global climate change. Currently, technology has allowed us to sequence the genomes not just of one individual and not of a dozen, but of hundreds. This allows us to see patterns in the data that reveal how evolution works. For over a century, theorists have been thinking about how this happens, but now we can see with real data how these patterns work and how evolution creates solutions to clear problems. To be perfectly honest, I do this because I find it fascinating to understand how evolution works. What are the fundamental rules that apply to all organisms? Happily, these rules apply to crops also, and they help us make food supply and security stronger in the future, facing changing climates and global volatility.
Welcome, Levi.
Thank you.
You're going to talk about whole genome duplication. If you were to explain what that is to someone without a background in biology, how would you describe it?
So whole genome duplication is a phenomenon that happens in all kingdoms. It's happened in our own evolutionary history, and it's particularly common in plants, especially the crops that we eat. The majority of them are whole genome duplicated. What does this mean? If you think about everything that makes the body plan that is you, or any organism for that matter, this DNA that makes chromosomes and together is an entire genome. And if you think about this suddenly being doubled in a single generation, this is whole genome duplication. Very simply put, it's a doubling of the entire blueprint that makes an organism. And this presents challenges for the cell, but it also presents opportunities. If you think about suddenly having twice as many instructions for different possibilities that you can draw off of and recombine, this is whole genome duplication.
Thank you.
Yeah.
And how does it differ from a typical mutation?
Yeah, it's commonly called the largest mutation, right? If you think about the common thinking of a mutation as a single letter change, A, T, G, or C in the genome. You had an A previously, but then suddenly it becomes a T. This is one letter out of your 3.2 billion letters. In fact, it's worth mentioning, you don't just have 3.2 billion or one genome in you, you actually have two, right? You have one from your mother, one from your father. And if you change one letter, that's one letter out of 6.4 billion. A whole genome duplication, that's 6.4 billion again, right? So a massive mutation, which then allows for much more variability to be held in your pocket.
So how does mutations and whole genome duplication complement each other?
Yeah, this is an excellent question to follow up on that, because I talk about suddenly the genome doubling, yeah, so what? I have twice as much of the same thing I had before. But over evolutionary time, even a couple of generations in a population, this allows more mutations to be held onto as a sort of masked standing variation, right? Those are technical population genetic terms, but they're perfectly understandable, right? You think about variation that stands in a population that is held within a pool of genes or a population's potential, right? And we're thinking here of crops as well, what can be held in that common reservoir to be used in changing environments or in volatile environments. These new mutations, they can be held more easily if the genome has duplicated, because you have the current copy that's being used, but you can hold on to the new mutation. So they complement each other pretty well.
Could you say something about what species? You mentioned crops or vegetable? No, plants. But what other species use this?
I do have a short list in the lecture, but it's worth noting that the majority of our crops are whole genome duplicated. So wheat, broccoli, cauliflower, maize. You name it almost. Think of a berry, grasses in general, many fish also, and even in our own evolutionary history, we've had whole genome duplications. We are the product of these things. So sugar, coffee, some of the favorites. Yeah, you name it.
Common. Yeah. Please tell us more.
Okay.
Yeah. I would like to introduce Levi Yant, professor of plant genetics, talking about the double-edged sword of whole genome duplication.
Thank you very much. So, thank you very much, everyone, for attending, and thank you so much to SLU for having such a pleasant several-day event. This is not common where I'm from, and I have to say it's really a wonderful experience. So as I've mentioned in the initial questions, whole genome duplication is this doubling of the blueprint that makes an organism. It's not something we talk about every day, but it actually underlies everything we eat. It underlies our own evolutionary history, and it underlies many very crucial health implications for humans, including some of the most aggressive cancers are whole genome duplicated. And it is the patterns that we observe in plants and other organisms that are well paralleled in human cancers as well. So it's something that impacts us in many different ways, even though it's not a part of everyday life. And today I'm going to be talking a little bit about the potential that whole genome duplication offers, but also about some early barriers to survival in whole genome duplicated organisms. So it's this kind of paradox in evolution. Early challenges and later gains. So when we talk about a genome, this is usually an abstract concept for people, a bunch of letters, right? And the way we study them, in fact, is both using pictures, cytological pictures on a microscope, and also these letters bioinformatically, primarily. What's pictured here is a genome from a common plant, a lab organism called Arabidopsis, and it has two genomes, like I mentioned, and you see these pairs of lines, right? So you've got these two touching, and they're dancing along together here. And I use dancing quite explicitly because everything that you see in the organism is choreographed by different proteins and processes that dynamically happen over time. And they must be orchestrated correctly, or the organism does not survive. Here you see these two pairing here, a couple here. There is an order in this picture. And this order creates beautiful organisms, right? So this is the Arabidopsis I mentioned. The plant molecular people out there will be a little disappointed because this one's a different species from our favorite lab rat plant. I call it Arabidopsis arenosa, the beautiful sister of Arabidopsis thaliana. And it's adapted to some very extreme environments, high alpine environments, salty beaches, toxic mines. It can handle quite a lot of challenges that the diploid sister doesn't do so well with. Of course, we use our lab organisms as tools to understand the molecular biology, that can be transferred back and forth. But it's these wild species that teach us things that molecular models have difficulty doing, because the wild ones are living in these extreme environments and adapted to them. So we study how it is that these exceptional organisms are able to tolerate poor soil, right? This one is the one I ate in the movie. Please don't yet ask me to pronounce this one. Scurvy grass in English. High in vitamin C, was used among sailors, probably Vikings, and certainly pirates, so I understand. You can make beer from it as well. So there is a future possibly here in cropping systems, I hope. But again, it's these exceptional organisms that we study because they can teach us things that both crops and lab models can't so easily, but can be used in conjunction with lab models to enrich crops to handle new challenges. So again, what is whole genome duplication? You can visualize it different ways. Here I'm just showing a couple of chromosomes hanging out in the cell, undergoing this chance event, which is happening continuously in the wild, and suddenly being duplicated. Again, this happens in a single generation when sex cells are made among parents, right? So there's a continuous production of these whole genome duplicated lineages that usually don't survive, but sometimes do. And it's the production of these things that we've taken advantage of over millennia for some of our core foodstuffs, from basic starches, grasses, berries, useful trees, right? Some of our most important trees are polyploid or whole genome duplicated. Fish, insects, many insects, bacteria, and even the most aggressive cancers in humans are polyploid. So gliomas commonly are polyploid, and they take advantage in the short term of their extra genetic material to adapt to the host and diversify. So this is a microevolutionary example of what I would say is a macroevolutionary process in plants that we see in cancer. And we see some of the identical genes being key to this. So again, 2 genomes, right? You now know how to look at it. Ugh. It looks like a malfunction, right? No, this is the struggle that's happening in the plant. How do you choreograph that? I'll get rid of this because it hurts the eyes a bit. But really, I'm talking about this early problem of managing the choreography of the cellular activities that blocks most of the survival outcomes, the positive outcomes of polyploids, but which we know well later allow for diversity generation, okay? And speciation. So this is a fundamental evolutionary question. How are new species formed? Quite commonly, genome duplication underlies this. But again, I keep talking about dancing. Okay, so here is the normal dance. I want to show you some raw data to give you a really clear, concrete understanding of what's happening. This is a set of pairs of chromosomes dancing in a circle, almost like a Matisse painting, right? And each set of those chromosomes is engaging in a crossover. And this is the reassortment of genetic material. I was married in Scotland. Polyploidy happens in a single generation, right? And if you think about this, the dance becomes quite different. And I came here from England. And if you were to imagine the same rate of crossovers occurring among twice as many partners, you can have entanglements and a mess then when the cell tries to divide. This is ubiquitous across life, right? This happens in most polyploids. But there are these rare hopeful monsters, as Darwin called them, which are able to then stabilize this dance reducing the number of interactions and allowing for cell division to occur in a ordered fashion again. And here's a COVID era selfie of my lab. And so what we do most of the time is we try to understand what these solutions are, right? And so I talked about pictures. This is the kind of thing we look at under the microscope. And then to understand the mechanistic basis of this, we look at the genomes and then study the function of what is found. I'd like to share with you then what happens in the lab to study the genomes. How do you do this, right? So here, we want to go from non-survival to survival. And so we compare what is it that these exceptional wild organisms do to stabilize. And we do that by a genetic association approach. This is the simplest kind of genetic association, right? So you may have heard the term GWAS or association study. This is simpler than that. It's simply looking at what's different in one group versus another, right? So if we were to imagine a population of individuals, each having a genome or 2 or 4, you line them up using current generation sequencing. And if you were to imagine a mutation suddenly occurring in that population, which is advantageous, and you can think about any kind of adaptation you like, right? So adaptation to a certain soil, adaptation to, whole genome duplication. Yeah, suddenly this changed cell environment. Over time, individuals with that mutation become more common, right? Over evolutionary time. No matter what happens in other parts of the genome near them, that variant is held onto and increases in frequency. And you then end up with a population which, in some circumstances, everybody has that new change, which then allows them to survive. So what we do in the lab is we take the individuals which are not adapted, unstable, and again, this goes not just for whole genome duplication, but for any kind of adaptation or any kind of evolutionary change you're interested in. And we compare the groups that are not adapted with the group that's adapted, and we just sum up the frequency of the mutation in each population and look at how common the new mutation is when you subtract one frequency versus another. So simple addition and subtraction. Now real data. Okay. If we look across the genome and we compare one group versus another, we'll see an elevation in this difference. And this is all thanks to incredibly rapid increases in DNA sequencing, right? Everything we do really is underpinned by this and is accelerating madly, right? Beyond what I would even say AI is accelerating madly. The ability to sequence in different ways and with increased efficiency is changing biology by the month. Here, you can see that there are bits along the genome which are more different in one group versus another. In this case, the adapted polyploids, which when you zoom in on, point to specific gene coding regions, so parts of the chromosome that then make one protein or another, that say, this might underlie what's happening. This is what's special about this individual, this adapted polyploid. And when we do this, we see indeed the very top of these lists of different regions are the proteins that make the DNA dance in a particular way and allow it to sort the DNA, that change the number of interactions and distribute it. And so it gives us an idea of how this very basic molecular change mediates survival of one species or another, and then the potential for new individuals to form, new different species to form. Okay, but that's only one case. And to really understand if something is a fundamental biological process, you need to look at separate independent cases of it. You need replication, right? And so when we do this, we found something quite surprising, which I think flies a bit in the face of some current thinking in evolutionary biology, or trendy thinking, I guess. And when we look at different whole genome duplicated species, what I'm showing here is a phylogeny or a family tree of one genome duplicated sister of a diploid or of a normal, another and another, here pictured as 3 different species, cousins, sisters, right? And we look at what processes are adapting. All of them have slightly better dancing, of course. This is essential for the cell to divide. DNA management, for example, in this beautiful sister of Arabidopsis is definitely the key process. It is the dead top of the list, and all of the most changed genes that we then biochemically test for function are indeed managing the dancing of the chromosomes back and forth. Not a lot else. But then others, the signal is looking more at is telling us that protein dosage is more important. That this doubling of the cell, which has been selected in major crops, larger plants, bigger output, because there's more DNA making more protein, this maybe isn't always the best thing for the cell. Maybe it needs to be knocked back down to a more normal level for that species. And so some species don't like getting bigger because they suddenly are making more protein. And then lastly, others are changing the way that they're transporting things around. And so evolution is a little bit predictable, but not so much, and that it has actually many different things to teach us, and it's been creative with what it has. And it's these natural solutions that we can then nudge our crops toward if we need certain specific tailored differences in the crops. And so I would profess that there's actually quite a lot that nature can teach us if we have the tools and the outlook to listen. And so that's what we do. So we go into these extreme environments. We find these cute little plants. This one's put everything into reproduction, right? Just this is the smallest plant and a long string of seeds. Living in a high UV, extreme environment, but taking advantage of the extra genomes, right? It's not a lot else growing up there. And it's actually taking advantage of that extra pocket I alluded to earlier, to hold on to extra genes and then to use them. So we have also this huge gene bank in polyploids that we can take advantage of, and that we've already identified some pretty exceptional and interesting genes and different processes for the cell, which can be bred into crops or engineered into crops. So what does this have to do with our crops? Here's an excellent example. So this is the humble potato. The humble potato is actually a polyploid. It's genome duplicated, but it's not adapted to its polyploid state. And it has a lot of trouble being fertile and producing seed set that's required. When I first, not being from a crop background, when I first learned that you had to put a potato in the ground to get a potato, there's a bit of adaptation happening in my own brain. I thought, where just put the seeds out? This is mad. Well, it's because it doesn't, the dancing of those chromosomes in potato, it's not so good. Some varieties have slightly better than others. And we now have identified in this Santrav, right, so Arabidopsis arenosa, the exact molecular changes that stabilize the exact problem potato has. And so we're now looking at different varieties of potato and seeing a gradation of stability in this chromosome dancing, and now going to associate that with different variants of those same genes we know from nature to stabilize potato. So this stands as an example of a very tangible and clear example where we can use natural solutions. And then lastly, one that we've already done quite a bit of work in collaboration with good friends in Spain is this sister species of Raps, right? This looks just like your Raps oil plant, but it's a recovering polyploid. So I've given this picture that there's an initial challenge and then a great diversification, but then why isn't everything high polyploid? Or why don't we have, when I say polyploid, I mean doubled genomes, tripled, quadruple, 8, 12, even higher among bacteria, 100 sometimes. Well, it doesn't last forever. And it leads to a diversification and a drop. This hop of genome doubling leads then eventually over evolutionary time to a dropping. And this is in part because of this accumulation of mutations, so this interaction of mutation and genome doubling, but then also fragmentation of the genome and diversification. This is what we have here, a sister of ROPS that can grow on salty coastlines, so also degraded soils. And it's changes that we found in this and identified the biochemical basis of this that we now want to investigate in crops as well. So with that, I would just say thank you for listening, and thank you again for the really lovely afternoon.
So we have time for one or two questions, and I will just start with one. What do you think will be the time frame of going from basic research to applied research in this field?
I think it could be quite quick, for example, in potato, right? Because we have identified the solution, and we now just need to test it in this species. Other species where there are different kinds of changes takes a little bit more time. But again, given really incredible advances in the ability to sequence entire populations of individuals, we can identify very quickly and then start applying. It's just a question of the applied side. And I'm on the fundamental side of finding the changes, but that's one of the most exciting things about being at SLU is that there are so many people around here that I can work with, and we've already formed some partnerships, for example, with the potato.
Yeah, interesting.
Yeah.
Welcome with your question.
Thank you, Levi, for interesting subject and lecture. You mentioned humans as an example of species where we had the genome duplication in our history, but today we are regarded as not quadruple genomes, but we still have a double set of genes.
Yeah.
How does the process goes from being quadruple to double genome type?
Yeah, this is a very vanguard question, actually, because it has been impossible to really observe this, also to catch it in the act. The diversification occurs, and the genome does become fragmented over millions of years, but we see only snapshots, right? Even herbaria genomics can only see snapshots from some hundreds of years. We do have some species like this RAPS sister, which has about 30% of the genes are present as duplicates. And it's a very random kind of process as far as we can tell. But now that we're able to look at many individuals and look at these footprints over many species and populations, we have some ideas that it has something to do with gene expression, the way that the genome folds. There may be more exposed regions than others that are associated with this fragmentation. But again, it's something that we don't know the rules of yet. And in fact, it's been something that I think even a few years ago it was almost hard to imagine how one would assess that. So yeah, very forward. Thank you.
I'm a bit curious about, you mentioned that biology is changing by the month. So I would like to hear your comments on the sort of the change of the subject, especially when it comes to data storage.
Yes.
How would it change? How much would we have to increase data storage here? And what would that mean for you as a biologist? Looking at the data, would you have to focus on some part of it, or learning to sort of ignore noise or whatever?
Yeah, I'm delighted.
What do you see?
Yeah, I'm delighted to hear that question because this is a hot topic. So over the last 10 years, we've been able to look with short sequencing reads, right? So maybe 150 letters, and pull down billions of them, right, from a machine. When I say by the month, I really mean by the month, because now we can take very long stretches of high accuracy letters, 15,000 or 500,000 even, and then put the genomes together based on those completely de novo. the bioinformatic demands are high, right? So using CPUs and GPU resources, and happily the national infrastructure is good. We need to use that national infrastructure, right? This is the problem. The data storage itself, it's not so bad, to tell the truth. We need to reasonably archive it and make it freely accessible immediately. But this is not something that is too much of an issue, but we do need to have our own storage, I believe, to be responsible stewards and not just to rely on other storages. But it's not something that's outside, I think, of common allocation for different parts of research. But I think it's wonderful that you asked that question, because it's something that, because it's changing quickly, people don't necessarily have their eye on. Yeah.
We have time for a short question and a short answer. The final question now.
Can you engineer genome duplication in plants?
Absolutely, and we do it all the time. Yeah.
Warm thank you. Yeah. Thank you very much. Det här var dagens sista föreläsning. Och vi vill tacka alla professorer som har föreläst och alla er som har lyssnat. Såklart, och tack för alla fina frågor som har kommit. Och så fint att bli berikad med så mycket kunskap. Allmänbildande verkligen. Så stort tack till er alla, och det var allt för idag.